So this will be the last part of the notes on biomolecules, which is about proteins and nucleic acids. Something you need to realize about these two kinds of molecules is that functional proteins and nucleic acids are generally pretty huge molecules made of hundreds or thousands, or in the case of nucleic acids, up to millions of monomers, and have multiple functions. And so we'll talk a little bit about the functions. We'll talk more about proteins and some of the basics about structure of nucleic acids, but we'll come back later on in Unit 5, I believe, and talk more about 5 and 6 and talk more about nucleic acid function. So we have here proteins, okay? Whether you realize it or not, proteins make up by far the largest component of cells. 50% or more of every cell is made up of, or the dry mass of every cell is made up of proteins of various kinds. There are lots and lots of different functions of proteins. There are like at least eight different ones. And you see some examples here of the different kinds of functions that you can have. We'll talk about a few of them, but you're going to want to spend some time when you're taking notes in your book from the chapter to spend some time on this section of the chapter learning about the different protein functions. And of course, some of those we'll talk more about later on as well, but just have a little bit more familiarity with the functions of proteins than we're going to get from this notes video. Make sure you spend the time in your book. One of the most important ones, of course, one of the major groups is enzymes. Enzymes are used for lots and lots of things, okay? This is one of the major groups of proteins in cells. Every reaction in your cells is catalyzed by enzymes. Each of the enzymes is specific to certain substrates, certain types of reactions. How this works is something that we'll spend more time about in Unit 3. Again, you might want to spend a little bit more time looking at the ways enzymes are controlled in the chapter while you're taking notes, but we will spend some more time talking about enzymes when we talk about cell energy a little bit later on in Unit 3. Polypeptides are what we call the polymers of the monomers of proteins, okay? Functional proteins generally are going to be composed of one or more polypeptides, and so when we just talk about one single string of unbranched string of amino acids, that's usually going to be a polypeptide. We won't worry too much about it in class, but that is a proper way to refer to them. Of course, proteins are made of amino acids. These are the monomers, and these are organic molecules that have a carboxyl group and an amino group attached to a central carbon with some kind of an R group, and the R is a variable group, okay? The side chains, and there are 20 different ones, and the R groups are what make them unique from each other, and also changing the properties of the amino acids as well. Here we have the 20 different amino acids. Notice in this particular chart, and you have a full chart in your book that shows all of these together. I split it up into a couple of parts to make it more easy to look at. So you have them categorized by their behavior. Okay, these ones over here, these 5, have electrically charged side chains. They're hydrophilic. 2 of them are acidic, and 3 of them are basic. Okay, so they're going to react in solutions like acids and bases do. These over here are these are hydrophilic with polar side chains. There's 6 of those there, and you can see the various things that are involved. Sometimes cysteine is classified as nonpolar. Cysteine's the only one that has a sulfhydryl group in it, and that's important for some of the protein structure stuff that we'll talk about more in activities that we'll do. And then the last group here, this last 9, are the one with nonpolar side chains. They are hydrophobic, okay? And you see most of them have like methyl groups. Here's something with a polar, it looks like almost like a nitrogen base on it, but definitely, I'm sorry, not polar, but nonpolar. So they're all nonpolar side groups that they have on there. And those R groups, the side chains, determine how they're going to help determine the shape of the eventual polypeptide, okay, because of their interactions with each other and with the environment they're in. Some of them have a more affinity for water, like the hydrophilic ones. All of these ones are hydrophilic, these 11, and then these 9 here are going to be hydrophobic. So you can see how in a watery environment, that is the cell, that some of those are going to kind of fold to the inside to stay away from water just because of their properties. Polypeptides are joined together by peptide bonds. A peptide bond is a bond that occurs between two amino acids, between the amino group, the carboxyl group of one amino acid and the amino group of another. Okay, and they're formed also by, guess what? Dehydration synthesis once again. I don't expect you to know a whole lot about this other than you should be able to recognize when you see a peptide bond, because you're going to see something like this, a carbon with a carbonyl group that is bonded to a nitrogen with a hydrogen on it. And there should be another carbonyl group coming up pretty soon. Proteins or polypeptides have they have their sidedness, okay? They have a unique sequence. On one end you have the N terminus, that's the amino end. The other end you have the carboxyl end, or the C terminus, okay? And you can see also that those are going to have different behaviors as well, because this one's acidic, this one's basic, and so they're going to be kind of attracted to each other or to other things that are oppositely charged from them. A functional protein is made up of one or more polypeptides, and it's going to be twisted and coiled and folded into a unique shape. There are various ways that we represent this in pictures. Here's a ribbon model that shows you the various kinds of structure, these helices here, and the folded back and forth pleated sheets that you see there in the protein. A space-filling model is going to show you more what the three-dimensional shape of the molecule is going to look like. But these are all models of the same. This is all called a wireframe model that shows the interactions of the side chains there. But you notice if you look at the outline there, they all have the same structure. This is the same molecule, same enzyme probably. And there's a groove right there where whatever the target molecule is that this works on is going to fit. If this is an enzyme, it would be something like the active site. Okay. The big thing is that the sequence of polypeptide, the sequence of the amino acids, leads to that three-dimensional shape, and the protein structure determines its function. If it's not shaped properly, it's not going to work properly, and that's a really important thing to consider. There are 4 levels of protein structure, okay? They have these things in common. All functional proteins have 3 levels of structure. They have the primary, which is that involves the sequence of amino acids, the specific sequence there. They have a secondary structure, which has to do with the hydrogen bonding of the peptide backbone. It causes the amino acids to either fold or twist like this. And they have a tertiary structure that has to do with the three-dimensional folding related to the interactions of those side chains with the environment that they live in and with each other. Some proteins also have a 4th level of structure, which is called the quaternary or quaternary, you sometimes hear it called, structure, that comes when you have a protein made up of 2 or more polypeptide chains. You have to have all 3, first, primary, secondary, and tertiary, exhibited for the protein to be functional. It's not functional at this level or this level. It's not until it gets to this level that it can be functional at all. And that's an important consideration. The primary structure is unique. It's that unique sequence of amino acids. A lot of times we call it like beads on a string. They're in a certain particular order. The sequence of the amino acids is the primary structure. The interaction of the chain with itself is going to lead to the 2 different kinds of secondary structure, either the beta pleated sheet or the alpha helix, okay? And it has to do with the coils and folds in the polypeptide chain. The tertiary structure, again, is the folding pattern due to side chain interactions. In other words, those R groups are going to interact with each other. Either maybe they'll be attracted to each other, maybe they'll be repelled by each other. Some are hydrophobic, some are hydrophilic, and the hydrophobic ones will have a tendency to be folded to the inside. The hydrophilic ones will be on the outside. And then finally, the quaternary structure for those that require multiple polypeptide chains would be the final stage in the structure of the protein. Now, protein structure can be affected by things other than just the amino acid sequence, okay? The physical and chemical conditions in the area where the protein exists can affect the way the protein is folded Okay, if the pH is wrong, if it's got a high salt concentration, if the temperature is wrong, all of those things can cause the protein to unravel or to change shape in some way. This is called denaturation. Okay, some denaturation is irreversible, like for instance, the white of an egg is made of proteins, mostly called albumins, and when you heat that too much, it's going to be like cooking an egg, and that the egg white is going to go from that clear jelly-like substance to white and opaque. That can't be reversed, okay? But then other kinds of changes in some proteins can be reversible. And so you have a denaturation that's occurring here, and then a renaturation when the conditions change back to the normal condition. For instance, if the pH is a little bit off, it might cause it to unravel a little bit, but if you correct the pH, then it'll go back to its normal shape, okay? If it's denatured, it's inactive. It's not going to work to do its job, and that's a really important concept to understand. Nucleic acids are in charge of storing, transmitting, and expressing hereditary information, okay? And you already know that somewhat. The amino acid sequence of a polypeptide is coded for by the sequence of bases in the DNA, and it's coded for by a particular sequence called a gene. Okay, genes are made of DNA, and the DNA is made of monomers called nucleotides. There are two types of nucleic acids. There's DNA, deoxyribonucleic acid, and RNA, which is ribonucleic acid, and they are both involved in the transmission and the inheritance and the expression of that information. DNA has directions for its own replication, and it also directs the synthesis of messenger RNA, which then directs the synthesis of proteins. And so those things are going to work together. We'll talk a lot more about that in Unit 5 and 6 when we get to that part, talking about how things work. Okay. The components of nucleic acids are called nucleotides. A polymer of nucleotides is called a polynucleotide. And the nucleotides are composed of three parts: a 5-carbon or a pentose sugar, a phosphate group, and one of the nitrogen bases. There are two categories of nitrogen bases, the pyrimidines and the purines. Okay. In DNA, we have deoxyribose sugar. In RNA, we have ribose sugar. If you look carefully at the structural formulas down here, you'll see deoxyribose, the D means remove, has one less oxygen on it than the ribose sugar, and that affects the way it bonds with the nitrogen bases. Okay. The phosphate group is attached to the number 5 carbon here. Remember, we counted the carbons. We numbered them from going clockwise from the O, from the oxygen. So here's 1, 2, 3, 4, 5. So on the 5' carbon, that's where the phosphate is going to attach. And that's going to attach to the 3' carbon of the next one up in the chain. And so we say that the structure of the DNA and RNA goes from 3' to 5'. Sorry about that. And the nitrogen bases, we have 3 purines. We have cytosine, which is found in DNA and RNA, thymine, which is only found in DNA, uracil, which is only found in RNA. These two are very similar to each other. One of them bonds with the ribose and one bonds with the deoxyribose. And the 2 purines are adenine and guanine. Notice that the purines have 2 rings in their structure, and the pyrimidines have only 1 ring. And you learned in your initial biology class, you know, that the A always bonds with T, and C always bonds with G when they're in DNA, and so they're complementary base pairs is what we call them, and we'll talk more about that again later on in Unit 5 and 6. Okay, RNA molecules are usually a single polypeptide chain, and the DNA have 2 polynucleotides that spiral around each other, running in opposite directions, forming a double helix. We call that arrangement anti-parallel because the 2 backbones run in opposite directions because of the way the sugars orient with each other. And then one DNA molecule includes lots and lots and lots of genes, on the order of, probably 1,000 or more genes on a DNA molecule. And DNA molecules are composed oftentimes of millions of base pairs, so they're very, very large molecules. And that ends the unit, or the notes on the unit 1 about the biomolecules.